Hi. Hello, hello. I see people starting to sign on. Hello, I'm going to go ahead and share my screen. Welcome. This is the webinar, Getting to Know Your Scientific Community. And I wanted to tell you a little bit about how the webinar is going to go as people are signing on. We have a Q&A section at the very bottom of our screen. And while our panelist is speaking, if you have any questions, you can go ahead and ask them while they're speaking, and it won't interrupt them. And we love when you ask questions. It really makes it fun and interactive. And I also wanted to make sure you knew that this is going to be a recorded webinar, and we will later be posting it on YouTube. So when you ask a question, I won't be saying your name. I'll just be saying, oh, we have another question. So just so you know that we really appreciate all those questions that you send in, and you can ask them at any time. I wanted to make sure that to welcome you. My name is Kirsty Bourne, and I'm going to be your host. Today's webinar is sponsored by my company, Elim Bio. Elim Bio is busy supporting researchers on the front lines, and we are proud to say we're helping scientists solve the problem of COVID-19. During this unusual time, we wanted to reach out and help feed future scientific curiosity, and we felt the best way to do that is to give you at-home students a chance to meet some really cool scientists that we know out in the scientific community. And we have a very cool scientist and panelist here today. I'd like to introduce you to Dr. Siddheshvar Bhela. Dr. Bhela is a postdoctoral researcher. He's a postdoctoral fellow at the University of Minnesota. And in case you don't know, a postdoctoral researcher or postdoc is a person who is professionally conducting research after the completion of their doctoral studies. Dr. Bhela's current research uses mouse models to explore and better understand immune responses in tumors and autoimmune diseases. He has different scientific research published 16 times, and he is a true T-cell expert. And now what you've been waiting for, I'd like to hand over the floor to Dr. Bhela. Could you tell us more about your research and your current scientific work?
Yep. Can everyone see me? And I'm going to see, can everyone see me?
Yep, we can see you. Perfect.
Okay. Hi, and thanks so much, Mrs. Bourne, for that introduction. I'm Sid, and like Mrs. Bourne said, I'm a postdoctoral fellow at the University of Minnesota. And I believe there were some webinars before where there was a scientist who talked to you about the immune system. So today I am actually going to talk to you a little bit more about the immune system and autoimmune diseases. So the immune system helps us protect from harmful germs such as bacteria, virus, fungi, and parasites. There are viruses such as the chicken pox or the flu that you get, bacteria that can cause pneumonia, and also strep throat. Those are all bacterial infections. Fungi such as like athlete's foot, where you get itchy rashes, and parasites that can infect and they can cause kind of like diarrhea. And all these things, they're present all around us, and our immune system is like kind of like the defense system of a country. And in the same way that you have different soldiers and different kind of players that protect the country, similarly the immune system has a lot of different players and different cells that help protect against these harmful germs. So how does the immune system kind of protect us from these germs? One of the most first barrier that these germs come across is your skin. So the skin is the kind of the largest organ of the body, and it prevents any of these harmful germs from getting inside your body. Not only the skin, but also you have your mucosal membranes. And these mucosal membranes, they secrete this mucus. And this mucus is like this thick protective layer that just kind of traps all these harmful bugs, and they don't allow them to get inside the body. So that's kind of like your first line of defense. The second line of defense, which is kind of if for some reason germs get inside your body, the second line of defense, which is not necessarily very specific, it will recognize something that is harmful, and it will decide to go and kind of eat it up. And we call these cells as these cells that have the ability to kind of eat up these harmful germs. They're called as phagocytes. And this here is just like, it's a clip that shows this phagocyte or this cell that is kind of chasing a bacteria. And if you see that it kind of chases it for a little while, and then finally it'll get to it and it'll kind of eat it up. So here you can kind of see how that cell is how that bacteria kind of gets eaten up by that cell. So this is kind of like your second line of defense, but there are more added layers. So this is just, if the bacteria still, or if the germs still do not get cleared, then you have your third line of defense, and these are your specialized cells. And these are the kind of cells that I have been very focused and kind of interested in studying, and kind of like Mrs. Bourne said, that I study T cells. So this is a clip of this green cell right here is kind of been attacked by a harmful germ. And so what this T cell, which is kind of not colored, will go and recognize this cell, and then it will poke holes into that cell. And when it pokes hole into the cell, it kills that cell. So this is kind of a very specific cell that can only do this, and it has the ability to recognize these cells that are of your body that have been kind of attacked by germs. And the best part about these T cells and B cells, I mean, B cells are also kind of, B cells are cells that they have these Y-shaped antibodies, and they secrete those when they see harmful germs. And they kind of, they also provide a protective layer and protect us from those germs from really causing much more damage than they've already done. The best part about T cells and B cells is that once they see these germs, they remember them. So they have memory. And so now when they see it the second time around, then they have this ability to fight them a lot quicker, and they will protect your body from those germs entering into your body. So imagine a situation when your immune cells attack your own body instead of protecting it. And this is what happens in case of autoimmune diseases or autoimmune disorders. And so what happens is your T cells, they can poke holes in your body's healthy cells, and they can kill them. And you don't want that. That is a bad thing. You don't want the T cells attacking your own body. And the B cells, they can produce these, like I told you that B cells can produce these Y-shaped antibodies. They can go and attach to a healthy cell and then flag them and let other cells and kind of alert other cells to kill that cell, because it says that by kind of attaching to that cell, it alerts those cells that this is bad and we need to kill it. So basically, we don't want these T and B cells to attack our own body. We want these T and B cells to help to protect us from germs such as bacteria and viruses, but we do not want them to attack our own body. But in autoimmune diseases, these T cells and B cells attack our body. And so just, there are hundreds and different types of autoimmune disorders. And these are just some common examples. And I think that some of you might have already heard these. There's Crohn's disease, which is also sometimes called inflammatory bowel disease. where your immune system, so your T cells kind of attack your digestive tract, and so that leads to inflammation in the intestine. Another example that you might have heard of is multiple sclerosis, where the immune system attacks the nerve coverings, and so it doesn't allow the nerves to function and really allow the nerves to send signals to your body. And so you have symptoms like weakness, and you have double vision. And so that's another autoimmune disorder that really does occur. Now, another one that is kind of common, and a lot of people kind of have this, and it's a rheumatoid arthritis. In this case, the T cells, they attack the joints, and so you have inflammation within the joints. Some other examples are like psoriasis. So in psoriasis, your immune system attacks the skin, and you get scaly rashes on your skin. And then there's type 1 diabetes that also is an autoimmune disease in which your immune system attacks the pancreas. So in all these cases, your T cells and B cells in some cases are attacking your own body, and it's attacking different organs. And when those T cells attack those organs fail, and you need those organs to really be healthy. So one big question is, where do these cells come from? Why does only some people get autoimmunity, not everyone? That question in itself scientists are trying to figure that out. It's a very complicated question. There are many different things that can cause autoimmune diseases. We ourselves don't quite fully understand this. And the other question is, can the body take precautions while the immune system is developing such that these autoreactive cells are not produced? So to kind of better understand this, I'm gonna just talk to you about so similar to like you guys, you go to school to get educated, and once you're educated, you make good decisions, and you kind of you learn a lot of things during this education. Similarly, T cells, before they come out into the body, they need to be educated. And the place where T cells get educated is actually the thymus. So thymus is a very fascinating organ. It's present between your lungs, and it's kind of it's in front of your trachea. And what it does is that it basically, if a T cell would normally attack the body, then what the thymus does is it would say that it would give it signals to tell it that you're a bad guy because you have the ability to attack the body. We are going to eliminate you. So the T cells kind of undergo this education process, and these cells that are harmful for your body are removed in the thymus. For the B cells, it's a little bit different. The B cells get educated in the bone marrow. And in the bone marrow, so the bone marrow is all over your body, and these B cells that would normally create these Y-shaped antibodies that would go and attack your body, they are normally, if they were to attack your own body, they would be eliminated in the bone marrow. And so the body has these different checkpoints. And so you have your T cells that are educated in the thymus, you have your B cells that are educated in the bone marrow. And we don't want something that would attack us to not be deleted or eliminated from them. But what happens in autoimmune diseases, so what happens when these T cells and B cells are not properly educated in the thymus and bone marrow, what happens is you have an autoimmune disease. So you'll have T cells and B cells that are now attacking your body because they were not eliminated in the thymus or the bone marrow. So how do I, as a scientist, study these autoimmune diseases? So we use a mouse model. So we have these mice. And if you look here, this is the small intestine of a healthy mouse. And if you can see here, you see that black stuff within the small intestine. That's the fecal matter. And if you notice that this is a healthy mouse, and this is what a normal intestine would look like. But if you induce an autoimmune disease in a mouse such that now your T cells are going and attacking your intestine, what happens? So this is the intestine for a mouse that has autoimmunity, and your T cells are going and attacking the intestine. So if you notice here that it doesn't have that black fecal matter, that's because what happens is because these mice have diarrhea, so all their fecal matter is liquid. And so you really can't see it. They have a lot of that stuff. It's just liquid, and so you don't see it. And then you can also see that in the mice that have this autoimmunity, the small intestine is kind of thicker. And so that's just kind of suggesting that there's a lot of these cells that are attacking it and kind of causing this inflammation. So why do we use mice for research? One of the reasons, I think that there are a lot of, you kids have seen a lot of different scientists, some that work with fruit flies, some that work with I mean, there are many different models that you can use, but why, as immunologists, do we love working with mice? Having said that, it's bad to use animals for research, but I think that in the end, we're trying to save lives. And so I try to, it's one of those reasons that, at the end of it, we're doing good. And so why we use mice for our research is because the mice their DNA is very similar, or their genome is very similar to humans. And so that's one advantage of using mice. The mice they're small, and they're very cost effective to keep. They multiply quickly, so they can reproduce as often as every 3 weeks. And they also have organs such as your heart, brain, lungs, kidneys, and also the immune system very similar to that of humans. So this gives us an opportunity to kind of use these mice to really know what is happening in human diseases. And mice, you can induce a lot of those diseases that happen in humans. You can kind of induce those in mice and kind of study what are the different things happening, and you can get more clues about how to solve the whole puzzle of how and why autoimmune diseases occur. And so in our lab, what are we trying to do? So like I said, that our lab mostly works on T cells. So B cells can also cause these autoimmune diseases, but we kind of focus on T cells. And our goal is that we want to, those T cells that did not get their proper education in the thymus, our goal is to basically somehow get rid of those T cells or suppress them so that they do not attack your body. And that will then help us prevent any autoimmunity that is occurring. And we do this by many different ways. We can induce these autoimmune diseases in mice and kind of study what happens if we can if we can somehow manipulate the immune system such that these mice no longer get or do not die from the autoimmune disease. So who are we really helping with our research? So our research will help us to better understand how these T cells kind of destroy the different organs in the body. And so essentially what our research will help generate a better understanding of how these T cells are attacking the body. And if we can figure that out, then we can design drugs to prevent those T cells from really attacking the body. We can manipulate the immune system somehow such that the T cells no longer attack the body. And that is kind of the overarching goal. And also, if we can do this, we can help a lot of people that have autoimmune diseases. So I think I'm going to talk to you about a little bit about what a typical day when I go into work looks like for me.
Can we do a couple questions before we do that part? Is that possible?
Sure. Yep.
Okay,
yep.
Wonderful. We had a question from someone and they said, what happens when people don't have a thymus? They have a family member that had heart surgery and it was removed in the process. So they're probably a little worried about them now that they know the point of the thymus.
So that is a great question. So the thymus actually is very critical when you're young. So when you're born, that's when your T cells are kind of getting, during that development phase, your T cells are getting educated. And if you see most adults, their thymus, so the thymus is kind of big when they're young. But when when kids start growing older and as you become adults, the thymus automatically becomes a lot smaller. So the thymus kind of, once you're an adult, it's not really playing that prominent of a role. It's more, the thymus main job is happening when you're a child, when you're very young, and that's when your T cell education is really happening.
Kind of like real school.
Yep.
All right, we have another question. Is the mouse a model for all autoimmune diseases or just Crohn's?
So that's a great question. Our particular lab specifically studies autoimmune diseases that happen in the small intestine, but there are a lot of mouse models available for a lot of different, there is a mouse model for multiple sclerosis. So not every autoimmune disease has a mouse model, but there are a lot of different mouse models for different autoimmune diseases. But there are also about 100 different autoimmune diseases. So it's hard to kind of have a model for each autoimmune disease, but there are a lot of models available out there, yes.
And how do you get a mouse with an autoimmune disease? Do you
So for us so that's a great question, and I think the best way I can answer that is we can actually take T cells that normally would be deleted, that would normally would be eliminated in the thymus. We can take those T cells and we can transfer them in the mouse. And when we transfer those T cells, they go and attack the organ. And so that's how we induce autoimmunity. So normally those mice do not have those T cells, but we have a way of generating those T cells and then transferring them into the mouse such that those T cells will then go and attack the tissue.
We had one of our youngest students ask a question. They were wondering, how many babies do mice typically have?
So that depends, but usually you can get up to 7 to 8 babies in each litter. And
do you let the mice go when you're done with them? What happens to them when you're done with the research?
So usually there'll always be some form of research. They'll keep continuing. So if I'm not there, someone else will take over the research and they'll still keep on using those. So the goal is that if we finally find a cure for, let's say, Crohn's, and that somehow we can really have a good modality of treatment, then we can probably start researching something else that still needs to be. But right now, there's a lot of gaps in knowledge to be able to design really good therapies for a lot of those autoimmune diseases that are there.
They just keep working with someone new.
Yep.
This is another question from a fifth grader. How do you know the difference between good T cells and bad T cells, or how does your body know the difference?
So normally your body is always equipped with good T cells because all the bad T cells are removed in the thymus. But sometimes with people that have autoimmunity, the bad T cells come into play, and it's very hard to only target the bad T cells because the T cells that are the good and the bad, they look very similar to each other. So it's very hard just to target only the bad ones. So ideally the best goal would be to only target the bad T cells so that the good T cells are still there to fight all the other harmful germs.
And we had another question that someone noticed that the T cells, they go to school in the thymus, and then the B cells go to school in the bone marrow, and they were wondering if that's how they got their name. That they were named.
Yes, actually, that is exactly how they got their name. Yeah, I forgot to mention that, but that's a very good observation. And yes, that is exactly how they got their name.
Wonderful.
Based on where they were educated.
And then let's see. Oh, sorry, that question, it was hard for me to follow. I'm going to read it and then come back to it. So with the phagocytes, we talked about how they kind of chase after the bacteria. How do they know where the bacteria is?
So the bacteria itself, they usually secrete these toxins while they're moving around. And the phagocytes have these receptors that can kind of recognize those toxins. And so they kind of just follow that toxins, and they kind of just follow and eventually eat them.
Wonderful. Okay, great to hear. Well, we'd love to hear a little bit about your lab and your typical day too. Would you mind showing us a little bit about that? I think you have a slide coming on.
Sure.
Great.
So I want to talk to you a little bit about what is the typical workday like for me. So a lot of my time goes into reading a lot of scientific articles, and kind of I read those articles, and then kind of I do a lot of planning and try to figure out what are the gaps that in knowledge that we don't know, and how can I really build those gaps, and kind of how can I better understand and help everyone better understand how the immune system is working, especially in the case of autoimmune diseases. And so a lot of what I do is thinking of new ideas, and then I set up a lot of experiments. I use mice, and I kind of induce autoimmunity in them by transferring these cells. And then once I transfer these cells and the mice get autoimmunity, then kind of I study the different tissues, and eventually I'll do a lot of data analysis that I get from these experiments. So it's kind of a circle where I do all these things, and then whatever data I end up getting, I'll use that to plan new experiments, and then kind of go back and doing this. And it's always something new. It's never, every day is something new. The best part about these experiments is I don't know what the results are going to be. So every time I set up an experiment, one of the most exciting things for me is to wait for the results. And I just sometimes it takes like 5 days for the mice to get autoimmunity, but I really want to know whether my experiment worked, if I was able to kind of somehow protect them from getting autoimmunity. So yeah, so it's kind of, these are some of the things that I do on a typical work day. And yeah. And so just to show you what a typical experiment looks like in the lab. So normally I transfer T cells into a mouse and induce autoimmunity. And so now after I induce the autoimmunity, I collect different tissues. So this here is your small intestine, which I showed you before. This is these intestines have a lot of cells that kind of go into the intestine, and then these small things here, these are your lymph nodes, and your lymph nodes are also kind of places where immune responses are kind of generated. And not only in lymph nodes, and this is spleen. So in order for your T cells to get into your intestine, they first need to go through the lymph nodes and the spleen, and then they enter into, they get activated, and then they enter into your small intestine. And so in a typical experiment, what I would do is induce these different, induce autoimmunity, collect these different tissues, and then look at the T cell responses in these different tissues. And I'm going to show you here, this is a clip from one of the mice that I had. And what I did is I took T cells and I put them into the mouse. And after 5 days, I took the mouse and I looked at these T cells, and this is your intestine. And you can kind of see these T cells moving around in the intestine, and they're kind of going, and they're actually destroying the intestine. They're attacking it. And so I can kind of study what, using these different techniques, I can really kind of study what these T cells are doing, where they're going, how are they attacking. So there's a lot of different things that I can do and understand. And once I have this information, I can design better ways to treat autoimmunity. Yep. So I'm gonna talk to you a little bit about, I'm gonna skip gears, and now I'm gonna talk to you a little bit about where my lab is. And so my lab is in Minnesota. So I believe most of you are in actually California. And I'm envious of you guys because the weather in California is absolutely beautiful all year round. Minnesota is not the best in winters. And so this is my lab here. And this is during summer, so you don't see any snow. And during summer, Minnesota is absolutely a gorgeous place, and everyone is out because after having 6 months of winter, no one wants to stay in the house. Everyone goes out, and the Minnesota is known as the land of 10,000 lakes. So we're surrounded by lakes, and so one of the most when summer comes, everyone just goes out. They'll be at the lakes, they'll be biking, there'll be all sorts of outdoor activities, and I absolutely love summers in Minnesota. What I don't like is the winters, and we sometimes get up to like 10, I mean, we get a lot of snow, and Minnesota is pretty good with clearing the snow after it's after snowfall, but still, the temperatures are really cold. Sometimes it goes down to -20 Fahrenheit, and so winter is something that I was not used to before. I'm not from Minnesota, but I've been there for about 3 years now. And it took me almost 1 year to kind of get used to the cold. And now I kind of know how to handle it. And I've started to enjoy some of those winter activities. And I also try to learn some skiing. And so I try to embrace the winter. And you have to if you're living in Minnesota. This is a picture of my lab. So from the inside, and you can see there's a lot of biohazard containers. So even though I don't necessarily work with any infections, like I don't work with harmful germs, but a lot of people in the lab, and even sometimes I myself end up infecting mice with viruses or bacteria. And so every lab is equipped with these biohazard containers and all kinds of protective equipment, which allows you to not be infected with any of these harmful germs. And so on the right, we wear these lab coats and we wear gloves, and we work on these biosafety cabinets. And essentially what these cabinets do is they kind of have this airflow, which prevents, if you're working with some kind of harmful germs, then it keeps it within the hood and it goes through a filter system such that it prevents it from coming outside in contact with us. So this way we can work with a lot of harmful germs and infect mice with them and still not have those harmful germs do anything harmful to or not attack us. So we make sure that we follow these guidelines so that we remain protected. Okay, so I'm gonna now, okay. So how did I get interested in science? So before I tell you about that, I'm gonna tell you, so I am from India. I grew up in the capital of India, Delhi. And I did my schooling till I was about, I did my schooling, and when I was 18, my parents, they sent me for a further education to US. And when I talk about, so for me, the journey of becoming interested in science was actually my parents. My parents both are, my mother is an English professor and my father, he's a scientist as well. And they always instilled in me the curiosity to ask questions about why things occur. And they always supported me in whatever goals that I wanted to, or whatever I wanted to do, they always supported me. And they're still living back in India, so I try to visit them as often as I can. So my journey started in India, where I was kind of interested in science, and I was interested in all kinds of science. I was interested in physics, chemistry, biology, but I didn't know what I wanted to do. I was kind of in this mixed bag. I was like, do I want to study biology? Do I want to study? So the best thing that I could come up with at that time was a biomedical engineering. So it kind of was a mix of biology, chemistry, and physics. And so for my undergraduate, I went to Texas A&M University and I did biomedical engineering. And I realized when I was doing biomedical engineering that I was kind of more attracted towards the biology. Like I kind of started falling in love with more the biology courses. And during this time, I used to really love this magazine. It's called Scientific American. Some of you might have actually heard of this. And I really used to love reading all the kind of current science that is being done in the world. And this was they do a really great job at simplifying it for non-scientists to understand. And so I kind of really got more and more excited, and some of these biology articles in there got me really interested in it. So and I didn't, at that point also, I did not know anything about immunology or the immune system. I just kind of had taken a few biology courses and I wasn't quite well versed with the immune system. What happened was, after I graduated, I kind of knew that I wanted to work in a lab that studies some biology lab that did some kind of research in it didn't have to be immunology, but it turned out that I ended up in a lab at Harvard, and I was in the lab of Dr. Claire Beecher Allen, and she was, I think I owe a lot to her, because she took me into her lab even though I did not know anything about immunology, and her lab was an immunology lab. she took me into her lab, and I learned a lot from her. And I also decided that since I did not know much about immunology, I used to go to work during the day, and then in the evening, there used to be immunology classes. And so I attended some of these classes in the evening. And the more I learned about immunology, the more addicted I got to it. And I then decided that I want to pursue my career in immunology. So I decided to get my doctorate in immunology. So I applied for graduate school. And if not a lot of you know, but I mean, graduate school for the most part, if you, depends on which field you go into, but for a lot of these biological fields, graduate school is free, and they actually pay you a certain amount of money to go to school. And so that was nice. And I got to kind of follow my passion of immunology. I got my doctorate in immunology. And then after that, I wanted to further my training and kind of get more independent, become more independent researcher. So I applied for a postdoctoral fellow, and I ended up in Minnesota in a lab that kind of studies these T cells and in autoimmune diseases. And I've been there for about 3 years now. So yeah, that's my journey about how I got interested in science. And I would say that for me, science is almost like an addiction. I really love it. I'm very curious, and I've always want to know more and more.
I agree. Science is pretty awesome. We have some other scientists that are on the call, future scientists, and they have some more questions for you.
Sure.
All right. It's really fun learning about your path and everyone's path to finding love of science. So I really appreciate you sharing that with us. Thank you. All right, so this question is more about your lab. They liked the picture of your lab and the stuff inside of it, and they were wondering, is stuff you do in your lab a secret, or do you share information that you find?
That's a great question. We love to share information because the more we share, the more we can collaborate with other scientists. So we sometimes don't have all the technology and the knowledge to do certain things. And if we share our work, then we can collaborate with other people, with other scientists, and use their technological know-how to really improve our own research. So I think that it is a very important thing to share science. And we try to do that as much as we can.
Yeah, I love that about science, how for the most part, people do try to share so that you can learn from someone else and they might have another idea.
Yep.
Pretty great. We had a question about the different ages of the mice. Do you ever look at the thymus in different aged mice, and do you have different results in the young and the old?
So I have not done that, but that's almost like whoever asked that question is thinking like a scientist. And that has already been done by other people. So they have looked at thymus at very young mice versus adult mice. And they have looked at the different differences in the thymus during both these stages.
Wonderful. I think we do have some scientists, future scientists on the, but they're probably already scientists. They just don't have the degree. What about your autoimmune diseases that you're looking at currently, and how are your results going? That was a question.
So currently, so in my current research, I've kind of found a way to suppress these T cells that would normally attack the body. And it's a unique way in which they do it. And I'm still in the process of completely understanding how that process works. And the goal is that eventually I can, once I know completely how this, I have this phenotype where I see that these cells are getting suppressed, but I don't completely understand how they're getting suppressed. So I'm trying to figure out, once I have the entire story about how they're getting suppressed, then I can maybe then the goal would be to kind of publish this work, and hopefully someone can take it further and maybe design drugs that can help people with autoimmune diseases.
Wonderful. Would you mind sharing a little bit about how publishing work happens for the students?
Sure. So publishing is so normally once I do all my experiments and I've found something interesting, and so what I tend to do is then I take all those results, just like a lab report. I would take all those results and make a big lab report kind of explaining exactly what I did and how I did it and what these results mean for the scientific community. And then I will take that lab report and I'll submit it to a journal. So for example, Scientific American is a journal, but we have our own science journals. And so I take that kind of lab report, submit it to a journal. Then what the journal does is it takes that paper or that lab report and it sends it to 2 or 3 other scientists that are well knowledged in the field in the field of immunology. And those other scientists will determine whether my work is complete and whether whatever I am claiming in my research is actually shown by the data that I'm showing. So if they think that this is not good enough and that I need to do more experiments, then it's a peer-reviewed system where they will then tell me that if you want to publish in this journal, then you have to do these further experiments in order for you to publish. Mm-hmm. And so it's a rigorous process, which is a great thing because otherwise anyone can just publish anything. But it is very important that we have these other scientists. And the best part is these other scientists, they do it voluntarily. They're not getting paid for this. They just do it because they love science. And so they will then review these particular lab reports and then tell you, hey, either you need to do more experiments to really claim what you're saying, or they think that this is really good the way it is, and then the journal will publish it.
Yeah, it's good for students to hear that even when you're adults, you have your work reviewed by other peers.
Yep.
A big part of making sure that you didn't forget something.
Yeah.
Yeah. We had a 5th grader that was wondering if you've ever gotten sick from your mice.
I have never gotten sick from my mice, thankfully, and that's probably because I follow all the guidelines and I wear all my protective equipment. If I did not, I might be susceptible to getting sick, but thankfully I'm not.
Good. And then we had another question. Just personally, they were wondering if you have ever known somebody with an autoimmune disease.
I do know one of my friend's mother actually has an autoimmune disease. She has a multiple sclerosis, and also one of my lab mates, he actually has multiple sclerosis. So I do know a couple of people that have it, and they are currently one of the things that most people with autoimmune diseases, they give anti-inflammatory drugs, and essentially that kind of suppresses your immune system and prevents it from attacking your body. But the problem, one of the biggest problem with these anti-inflammatory drugs is that it suppresses your entire immune system. So now, even though it's protecting your T cells from attacking your body, but it's also destroying all those cells that normally are good and they would normally kill the harmful germs. So if you are on these anti-inflammatory drugs, then you become much more susceptible to attacks by harmful germs.
Wonderful.
So that's why it is important for us to kind of, so like someone said, one of the students asked this question before, can we specifically target the bad guys? That's the eventual goal. If we can do that, then we can we can prevent a lot of people from having side effects from drugs.
Yeah, that would be great. More personalized medicine.
Yep.
Yeah. We had a question from another person about the model that you're using, the mouse model.
Uh-huh.
Would it be faster to use a different model, one that was closer genetically to humans, or is the mouse close enough for the research that you're doing?
So you can definitely you can use other models. People do use hamsters and the reason that we use mice is just because it is a convenient system, and a lot of reagents and are designed for us to be able to work with mice, and not, so if you wanted to work with other organism, then there is a lack of availability of reagents that we can use. And so we can definitely use other models, but I think that if we want to really delve deep into the problem and kind of really go deep into it, then the mouse is a really great system for
Wonderful.
it.
We had someone who saw mice being used in a movie that was about science. A famous movie, it has Will Smith in it. And the mice were very angry. They were wondering if the mice you're working with are angry, or are they generally happy when they're in the cage?
So mice are usually, they're very happy, and if you do not they're very happy, and they don't necessarily, the male mice are more aggressive than the female mice, but so most of the people tend to work with female mice instead of male mice, just because they're a little bit harder to deal with. The male mice, they're more aggressive, but other than that, they're very happy and they're very sweet and cute. And sometimes it's hard for me to even do experiments on them because I feel bad for them.
They are pretty cute. We have a lot of field mice in our backyard. They're very tiny. People think big mice, but really a lot of mice are very small. Well, I think that's most of our questions, and I don't want to take too much of your time. We've had a lot of really, really good questions from our attendees.
Yes.
We want to thank everybody for very intelligent and thoughtful questions. And I also wanted to thank you very much, Dr. Bhela. I really appreciate you sharing your time, your expertise, your knowledge. I know you really care about all the students in our community, and you've inspired many students to be future scientists today. And I wanted to thank you for your time and sharing.
Thank you.
Oh.
Yeah.
Wonderful. Well, friends, we have another webinar next week. I'm going to tell you about it a little bit right now. Usually I send an email on Monday, but Monday is Memorial Day, and I hope you guys will all be home celebrating Memorial Day with your family and thinking of those who died in military service and enjoying the people that you love. So I will be sending an email on Tuesday, but our next webinar is actually going to be on Thursday, May 28th, and it's going to be at 1 Pacific time again. And our panelist is Stephen Sakma, and he's from Sanford Berman Preeves Medical Discovery Institute. And I just wanted to thank you all for being here today. We had a wonderful time, and it was great to hear all your questions. And thank you again, Dr. Bhela. We really appreciate it.
Thank you. It was really nice, and I hope that you guys learned something, and I hope you become scientists.
Yes, yes. Stay curious, friends, and wash your hands too. All right. Thanks. Have a great day. Bye-bye, everybody.